So today I'm conversing with Mahendra Rao. He's a developmental neurobiologist and an expert on stem cells, not only neural stem cells, but other types of stem cells. And today he's going to talk about his work on stem cells and the current state of research in terms of translating the promise of stem cells, which has actually been a long time now, and a number of decades to the clinic, and it's been a slow process, and he can talk about the reasons why. There's a lot of technical issues that have to be addressed. Mahendra is currently the CEO of Implant Therapeutics at Pancella, a company. He's the former director of the NIH Center for Regenerative Medicine, and he's been involved with a lot of different companies that are trying to work on stem cells, including life sciences technology, which is like a major company. He got his PhD at Caltech and in developmental neurobiology. He did a postdoc at Case Western Reserve. Then he was a professor at University of Utah Medical School. Mahendra, you were born in India. Do you want to briefly talk about your upbringing and at what point you got interested in science, neuroscience, and then stem cells?
Happy to, Mark. Really good to talk to you again after working with you at the National Institute of Aging. I grew up I was born in a really small town in India. So I grew up in what people might think is America 100 years ago. But we moved to Mumbai, which is a pretty technologically developed city. And I did all my schooling in Bombay or Mumbai. I decided I wanted to go to medical school, and I was fortunate enough to get into medical school. And I was always interested in the brain and memory and learning. And so I worked to get a fellowship in neurology. And I was working as a neurologist, and I found it to be really frustrating in the sense that it was great. I could make a diagnosis, I could identify a rare disease, but there was not much I could do with it, 35, 40 years ago. And so at that time, I decided I wanted to do research. And I wanted to do research in neuroscience rather than doing studies in clinical trials. And so that's when I applied for admission to a neurobiology program at Caltech. And I was lucky enough that they took somebody who had no research background for a PhD program. And as you heard from Mark, the rest is I've stayed in neurobiology and neuroscience and looking at how one can make a difference because of my medical background. And that's what's led me to stem cell biology and repair and regeneration.
Now, your PhD and postdoc work, you were studying cultured neurons from the autonomic nervous system. And that's a sympathetic nervous system, for example, parasympathetic. And you were looking at how different trophic factors affect them. Is that right?
Yeah, it was a very fundamental developmental biology question, really, is how do you acquire the versatility and different forms of neurons that you see in the human brain when you're starting from very simple, what look like generic neurons. And so there was a model system that had been developed where it was very clear that you started off as a neuron with a particular neurotransmitter phenotype, and then that got switched during development from a completely different neurotransmitter program. So cholinergic to adrenergic and vice versa. And so that seemed like a wonderful model to understand how you can instruct cells to change their form. And that's what I worked on. And we helped identify a factor called ciliary neurotrophic factor and a cholinergic differentiation factor, which could cause this kind of fate change or transdifferentiation.
Yeah. And then when did you started working on stem cells at Utah? Is that right?
Yeah. So, you're a developmental neurobiologist and you're looking at how fate changes and how do you direct differentiation to acquire the final phenotype. And in neuroscience, it seemed to be keep going further and further back. So we went from looking at neurons to neural progenitors to neural stem cells, finally to the earliest stem cell, which was the embryonic stem cell at that time. And that time there was a lot of excitement because that was the time when that discovery had just been made. And I was fortunate enough that Utah actually, surprisingly, was still a big center for that kind of embryonic stem cell research. And I was contacted by a company called Geron, which led some of the work on embryonic stem cells. And that's how I got to working with early stem cell populations.
And so embryonic stem cells are, as the name implies, the stem cells very early in development after the eggs fertilize, then you get the early cell divisions. And then as development proceeds and tissues start to form, stem cells become more restricted in what, final type of cell they can produce. And so you identified in regards to brain development, restricted precursor cells. That is, they're cells that they can't form both neurons and glia, but they can only form one. And can you kind of summarize that work?
Yeah. So, a lot of what we've learned about stem cells has come from the hematopoietic system, where people have been pioneers in identifying a stem cell population called the bone marrow stem cell or the hematopoietic stem cell. And they developed this really nice model of how things develop. And it's never always through a direct stem cell differentiating into final cell types, but for a variety of biological reasons, it's important to go through intermediate steps. And nobody knew whether this was true in any of the other organ systems that was there. And at the time when I started this work, that was a big area of research. Is it the hematopoietic stem cell model that's true in other organs or systems, or is it some different model? And it turned out in the nervous system, it was very similar to the hematopoietic stem cell, that you had these intermediate and more restricted precursors. And my lab was among the first to identify several of these restricted precursors. There's a neuronal specific restricted precursor which can make multiple kinds of neurons. There's a glial precursor which can make both the two major glial types present in the nervous system, oligodendrocytes and astrocytes. And then there is an even more restricted precursor which makes only oligodendrocytes in the nervous system. And we were luckily able to show the hierarchical relationship between all of them, which was one advantage compared to the hematopoietic system, because we could do this in culture where they have had really a lot of difficulty in growing the hematopoietic stem cell in culture.
And then there are these restricted precursors, for example, say a neuronal precursor cell, it has certain molecular markers. There are certain genes that it expresses that, say, a glial restricted precursor doesn't. Is that right?
That's absolutely right. So it turns out that these fate specifications are imprinted onto the cell and they don't change. So you have that memory, and we can identify markers or a set of markers which uniquely identify these cells and follow them in culture or during development in vivo.
And this becomes very important as our conversation proceeds, because it turns out, early in stem cell research, so you mentioned the embryonic stem cells, the thinking was you could just take an embryonic stem cell, say, put it in the brain of someone who had a stroke and whose some neurons had dies, and that embryonic stem cell is going to replace the neurons. Unfortunately, it's not that simple, right? And so that's where these restricted precursors become very important, I think, as far as translation.
Yeah, Mark, you make a very important point, right? And I joke about it as I say that stem cells are not smart, right? I mean, they respond to signals to do what they have to do. And unfortunately, the adult brain doesn't have the same signals telling an embryonic stem cell to become the damaged neuron. While these restricted precursors, because they are present in the adult at the right stage, at least the signals directing them to differentiate are present. And that was the important finding. The inability to repair was because we had a reduced number of stem cells in the adult of these restricted precursors and these restricted progenitors, rather than there not being signals or the ability in the environment to be able to regenerate. And that's been a really important finding, which has led to all this translational work that we do.
And then there was a very important finding by a Japanese scientist in his group, Yamanaka, where he discovered you can you can take, say, fibroblasts from a person's skin, put them in culture, and turn them into cells that are kind of like embryonic stem cells, and that they're capable of forming different cell types. And that's called induced pluripotent stem cells. And that also becomes very important when you think of therapeutics, because in theory, you could take say a patient had some neurodegenerative disorder, you could take the fibroblasts from that patient, turn them into these pluripotent stem cells, and then in theory, convert them into, say, more restricted stem cells, or even maybe immature neurons, and then transplant.
Yeah, you're absolutely right. So, when we do organ transplants or we do cell transplants, there are 2 or 3 things that are an issue. One we already alluded to, which is, look, you got to get the right cell type, which has to be able to divide and respond to the environment appropriately. And that's why you need a partially differentiated or a fully differentiated cell type there. The second big piece has always been you have the immune system protecting you from foreign tissue or foreign invaders. And so you have rejection. And induced pluripotent stem cells gave you this ability perhaps to be able to solve both of those problems, because you could take your cell, make it into a embryonic stem cell like cell, the induced pluripotent cell. So it was your personalized cell, which would have your same immune family response. And now you could differentiate it into the more restricted progenitor or final cell so that you could transplant it, or you could make it as an organ in culture with your cells so that you'd have your organ or your tissue that you could transplant. And that was the real holy grail, that, if you will, but you had in one stroke solved 2 problems. And that's why Shinya Yamanaka got the Nobel Prize in a very short period of time, because the actual experiment is not that difficult, right? But the idea and the showing it and the problem that you solve is so huge that it was just very, very exciting.
Now that was it when you were at NIA, where I was. At that time, the induced pluripotent stem cells, that technology hadn't been established. This was in the early 2000, like, well, I don't know, 2001 or something, yes. 2002. And so you were working with human embryonic stem cells, and you got in a bad, from a science standpoint, got in a bad situation because so George Bush came in to be president, and the NIH essentially stopped all research on human embryonic stem cells, right?
Yes.
For a while. And that really hindered, because you were working on them. That hindered you. And actually, that was one of the reasons you ended up leaving. I think you just couldn't work, so you went to a company where you could work on them. But then the induced pluripotent stem cells came on board, so now it's actually better in a lot of ways than human embryonic stem cells. And then you got recruited back to the NIH to be director for what was then, I think, a new regenerative medicine center. Do you want to talk about your experience there? Because you made a lot of progress towards actually your own work, and then working with many other investigators, clinical, basic research, and translation. And that was actually a pretty exciting time in a way, because you're getting closer to the clinic.
No, Mark, that was, so you summarized it right. I joined the NIA and worked with you because of 2 very specific things that were happening. One was, at that time, there was a lot of excitement with working on embryonic stem cells. And before the change in the government, there was a lot of support for being able to do it. And then there was another thing that the NIA had invested in, which was the counterpart of this, which was large-scale human sequencing data, right? And they had a big plan with the Solexa, the pioneer of what we now look at for all the companies which are doing next generation sequencing, which had been developed. And we had some of the earliest steps to be able to do it. So you had the first knowledge of cells, and the way to be able to analyze them on a large scale. And that was a really very productive time. And so it was like incredibly frustrating that one had to stop doing that work because of political issues rather than any scientific issues. And that's where I decided to join a company. And the company continued to work on this along with working on induced pluripotent stem cells. So we were one of the leaders as a company in working on this stuff. And then when Francis Collins came on board, he realized that this was such an important field and that the NIH should make an important contribution to that. And so he asked me to see if I would be interested in leading this center that was set up. And we were very excited to do that because what Dr. Collins said at that time was, we don't want you to just run a lab. We want you to see how the NIH can become the center of providing, because we have dual functions at the NIH, not just supporting research in the intramural program, but also being able to make resources and reagents available across the world, not just in the United States. And so that was the program that I was tasked with setting up. And that's, as you can see, as you pointed out, the NIH went through a whole data accumulation process where we identified the needs. We went ahead and made sure that there were facilities and resources available in the intramural side so that we could make hundreds of iPSC lines from patient-specific lines, et cetera, for discovery and search, which has become a valuable resource. And then he had tasked us from making it also available worldwide. And so we set up this center so that could distribute lines that had been made without all these company restrictions that could be used by people all over the world. And they've become really some of the most important lines that people use around the world. And in addition, for translation, he said, I want you to identify the first 3 or 4 ideas which can use stem cells, and I want you to make sure that you can enable them. And so that's what was done. And as you know, the NIH has been one of the leaders in running iPSC-derived retinal pigment epithelia clinical trial study where they've already treated patients, and they have got an approval to be able to do that. But in addition, he asked us to make a GMP clinically qualified line that could be used for therapy. And that was the first line made in the world, thanks to the NIH and thanks to the support and vision of the NIH in being able to get that done. And that line is being used for clinical trials and early studies by at least 18 different companies. So you can see how the NIH made a huge difference. And I was really proud to be a part of that work to be able to get that done.
So with the induced pluripotent stem cells, so there's a huge amount of work going on this. And the idea is, say you're a patient that has a certain genetic disorder that's inherited. Could be Huntington's disease, it could be sickle cell anemia, doesn't matter. And so the idea is you can make iPS cells from skin cells from that person, and then you put them in culture, they're in culture, and then you figure out, how to direct them to the proper end cell. And Huntington's disease would be a neuron, maybe even a particular type of neuron, GABAergic interneuron, for example, which degenerate early in Huntington's, or in some other disease. It could be a heart cell, it could be a liver cell, whatever. And then you can study, you can discover, or at least work to discover why that gene mutation is causing the disease. And and we did some work in my lab on that with certain mutations that cause early onset Alzheimer's disease, and made some valuable findings, I think, that contribute to the understanding of that disease. Now, why don't you talk about so I think the thing to talk about next in terms of translation is actually what's done, all the hurdles you have to cross in translation to get some stem cell into a patient.
So, yes. So, there are 2 types of hurdles that are really important to keep in mind. One is the scientific hurdle, right? And the other is the regulatory hurdle that you have to make sure is important. So in the science hurdle, the standard problems of using cell-based therapy are really what we've already talked about, which is you have to know that you can take a cell and differentiate into the right final product and you can manufacture it, right? So that can be done. And the other is you have to make sure that there's no contamination of dividing cells so that you have no tumorigenicity and you can solve that problem in doing the manufacturing. So that there's been a lot of advance and you can do that. So now we can make cardiomyocytes, we can make natural killer cells, we can make macrophages, we can make dopaminergic neurons, we can make pancreatic islets, all of that at clinical scale for use in patients, and patients have already gotten cells from here to be able to do it. So we know that we can solve some of those problems. Some of them we can't solve. For example, we can't make platelets at an economical scale from iPSC just because of the numbers that we need are so large. Or liver cells, we know how to make them, but we can't make them at the right price, at the right cost for it to be good enough to be able to use. So scientifically, lots of work, lots of excitement, lots of companies, and they can do this. The second part really is, can we do this and the regulators feel comfortable that this is going to be safe? And that has been an equally difficult path because it's a new learning and it's a new methodology. So that regulators have also to figure out how to regulate it. So the one big problem is, if you have a pluripotent stem cell as a contaminant in your culture and you transplant it into humans, then you might get a tumor. And that if that's the case, then you can't take the cells out. Then what do you do? So please develop something to make sure that it's safe enough, or the risk-reward ratio is such that you will be allowed to use it. So that's a huge problem. Then the second problem is, a lot of times when you do all of these therapies with cells, it takes time. And humans live for a long period of time. So how do you design the clinical trial so that you know that these things which are going to be implanted and are going to survive, unlike drugs, which last for 24 hours or 48 hours, will be okay? And so the regulators had to figure all of those things out. How will you monitor? Is it the lifespan of the individual before we will approve a product, etc.? Right? So that's become a really difficult piece. Luckily, the FDA has still been a leader in the world in trying to figure out how to do these things. And we look to the FDA worldwide as how best to move this forward. So they've been slow, but they've been supportive in trying to get that done. But these are the problems, Can I do it? Can I do it at the right cost? And can I do it so that I can satisfy the regulators that this is a safe and effective therapy? And that's been really difficult to be able to get done. But we're getting closer, And I always try to remind people that this was the same thing that was true for monoclonal antibodies as therapy. Now the largest number of new drugs have been monoclonal antibodies for treating a large variety of things. But for 30 years, it was the FDA grappling with the safety, the side effects, having to halt trials, worrying about how you can make them, can we manufacture them at the right scale without having any of these sort of contaminants that are needed. So it's a very similar problem, but it's a scale and scope has become bigger with cells compared to even antibodies or other biologicals.
Yeah, the antibodies is interesting. So intravenous immunoglobulin has been used in the clinic. What's it for? I can't even remember what disease it is.
Yeah.
And
malaria, hepatitis, etc. It's all been used, right?
And that's been used in, I don't know, tens of thousands of people, and the safety seems to be good. So that essentially the way that works is you get a big pool of blood from a lot of people, and then you pull out the antibodies.
Yeah, the IgG fraction.
The IgG. And so that's safe. So you'd think, boy, if that's fairly safe, then you'd think one monoclonal antibody would be fairly safe as far as side effects. But you're right. I mean, one of the most common treatments for a lot of inflammatory disorders now is TNF, tumor necrosis factor antibodies. And even COVID, right? That's short term, but
neutralizing antibodies
from Regeneron, actually, which is a company you and I know about from a long time, because they did a lot of work on neurotrophic factors and BDNF and so on. And So why don't you talk about the kind of the well-controlled, thoughtful, regulated approach, which is the right approach to potential stem cell therapies, and then the quacks, because there's, so for example, if you have knee osteoarthritis, there's places you can go, actually a lot of places I think now, and they'll inject some sort of stem cells, I guess mesenchymal stem cells. Uh, could you talk about mesenchymal stem cells? What are they, and why are they being given to people for certain conditions? And do we know whether in any cases they actually have a real benefit?
Yeah. So, I have very mixed feelings about mesenchymal stem cells because it's really something where there's a very good scientific basis, but how it's been co-opted and misused is a sort of depressing example of how humans can have success in both spheres of activity, right? So the idea of, if you imagine our body has different repair processes for different types of injuries, right? Depending on where the injury has occurred, etc. So you've got skin, it's your skin cells which respond to repair. And then normally that repair occurs with the local cells that are there. So keratinocytes and epidermal cells will repair. If it's any organ, then it might be the liver cell, and it might be a stem cell if the damage is a lot, because then the stem cells get reactivated, much like what you can expect in any other system. So the idea was that, look, mesenchyme is the intermediate sort of connective tissue, and some cell has to make it. And so it was the mesenchymal cell. And then people realized that, mesenchyme turnover in the body is a really large turnover. So the cell should have a capacity to divide and should be able to make a lot of the mesenchyme or connective tissue. And long time ago, it was identified as a mesenchymal stem cell that's present in the adult. And unlike most of the other cells in our body, because of how much repair it had to do, it divided a lot more than the average adult cell. Now, so that part is all true. And then because it's connective tissue, a mesenchymal cell can make most of the connective tissue. So smooth muscle, tendons, little bit of skeletal muscle of a one particular kind, right? And most importantly, bone, and cartilage. So here you had a normal cell which could have these repair properties and that could do that. And everybody was very excited because this is its normal function. So heck, we could get a lot of these cells and we should be able to use them. And the jump here was the same thing that we talked about before is, look, I can identify them, I can isolate them, I can do this all easily, and I can take it from the same person. So I should just be able to do it. And I don't need to do any clinical trials. This is just a doctor doing that work. But any study or anytime you use something new, you could have a beneficial effect, but you know that it's placebo effect. There could be one of a kind effect, or you've not controlled the experiment, so you don't know whether it'll be repeatable. But people ignored all of that in the excitement of all of this. And then when companies got involved, they didn't want to do the studies. They just wanted to sell the product, right? So we had this whole unregulated piece that happened for a long time, and it was just misuse or overselling things to people, much like you do with anything else. And then that's where the FDA stepped in and said, look, we see a promise here. We understand that, but we're not going to have this all happen without thinking of it as an approved product. So they made it sure that this would be considered a drug. And if it is a drug, then it has to be regulated much like any cellular biologic or anything else as a therapy. Now, I'm going to give you the two extremes. So there's been a lot of hype with all of this stuff that's been done, and people have been using mesenchymal cells, and there are these little clinics. I remember I went for a trip down to Patagonia, and there is this little town which is called the town at the end of the world, right? Terra El Mundo. And right there, I see this little stem cell clinic advertising mesenchymal stem cells for pain therapy, right? So you realize that this has been something that's happened. But on the other hand, after clinical trials have been done with mesenchymal cells for very specified treatments, there are approved products in several parts of the world. And there are companies which are billion-dollar companies because they've been able to commercialize this product for its use. So Mesoblast is Osiris, a Maryland-based company, it turned out to be the first successful company to be able to do it. There's some company called Pluristyx. There's a company I was involved with in India called Stempeutics, which had the first mesenchymal product in India that was approved by the regulatory authorities to do it. So that's what's happened is once you regulate, you get uses, and you know what the extent of the use is. And so now we know that mesenchymal cells work. It's not as exciting, and the hype that people had is not as good, but for certain specific indications, they are very beneficial, you know? And so for treatment of osteoarthritis of the knee, rheumatoid arthritis now of the knee, some wound healing examples that have been there are approved products. Treatment of the spine, there is an approved product that I think is clearly beneficial, you know. But this thing about we will solve cerebral palsy or, we'll regrow the liver, which has been also hyped, has turned out not to be true.
Yeah. So are mesenchymal stem cells, would you look at them as similar to, say, a neuron-restricted precursor? Are they, in other words, you mentioned that they can give rise to bone, tendon, muscle, but they can they give rise to neurons?
No.
No.
So people initially thought that they were like pluripotent stem cells, and it turned out it's not true, right? But they can give rise to bone, cartilage, and tendons. So they are a restricted progenitor, or a restricted stem cell or a tissue-specific stem cell, depending on how you want to classify it. And in fact, now there's an argument in the mesenchymal field on either calling them a stem cell as opposed to calling them a progenitor or a tissue-specific stem cell in some fashion to be able to do it. But yes, they are like a neuron-restricted precursor.
Okay, well, so that's kind of encouraging from kind of proof of principle for one type of restricted stem cell. And you've been working and even are involved, I understand, in clinical trial of some glial-restricted precursors in ALS, Lou Gehrig's disease.
That's right.
You want to kind of briefly describe what's going on in ALS, and then you collaborate with Jeff Rothstein, right, at Johns Hopkins,
and Target ALS, which
yeah, and so, and his findings a long time ago concerning the potential role for astrocytes in helping protect the motor neurons that degenerate in ALS. So anyway, talk about ALS and your efforts there with your collaborators.
So, just as we talked a little bit earlier, for therapy, the CNS has become a very difficult site for therapy because of the innate structure of the nervous system. As you know, the neurons are not just present in one site. They have axons which go for, feet, several feet, right? To go from the top of the head to the spinal cord, for example. And they are integrated in a very complex network. So it's not just putting new neurons in, they can't make the right connections. And these connections are now much larger than they would be. And the rate at which neuron axons grow is very slow. So even if you could do all of this, it would take a very long time for the neuron to make all the right connections. So that has restricted us from what we can do in the nervous system, even though we can make all of these neurons. So in ALS, then that was why Jeff Rothstein and our findings and etc. were considered important, was it was clear that the astrocytes were also involved in the disease. And if you had healthy astrocytes, they could protect the damaged neurons. So you weren't curing the disease, but you were protecting the damaged neurons so that they would last for much longer, and you would delay all the side effects of the disease. And then that would really help with the survival and quality of life of patients. And astrocytes, unlike neurons, don't make these really long connections, and they can migrate in the adult nervous system. They can migrate for centimeters in a relatively short period of time. So it meant that you could really transfer the cells to be able to do this. And so we did the early experiments in animals, and we did some of the first work in humans in a trial to be able to do this. And you can transplant astrocytes, you get measurable beneficial effects. The effect is as we predicted. It doesn't cure the disease, but it prolongs the survival of the damaged neurons significantly so that you have long-term survival of animals to be able to do it. And then Steve Goldman did similar work in another disease, which is a demyelination model. And he showed essentially the same thing happening with the myelination with the oligoprecursor. in a demyelination disorder that you can do that with. So that was very exciting. I'm sorry to say, though, that it's been a very tough road because of the number of failures we've had in clinical trials in the CNS. So many of the big pharmaceutical companies have cut down their research and funding for clinical trials with novel therapy. And so there was this company I was involved with, which was called Q Therapeutics, which has FDA approval to run the clinical trials. And we haven't been able to get funding because of this limitation in funding in the CNS. Because, often diseases, it's not a huge potential profit margin always, right? In these things, you do these because it's an important goal and you will help cure the disease and it will provide benefit, even if it's for a small number of patients. But we can't all expect companies to be completely altruistic either. So
I guess maybe need to do a ice bucket challenge fundraising
yes,
thing.
yes.
Now, this ALS is interesting. So Jeff Rothstein, a long time ago, had evidence, this is like more than 20 years ago, evidence that the motor neurons, the ones that innervate your muscles from your spinal cord, part of the reason they die is they're being excited too much by the neurotransmitter glutamate. Yes. When they're under a lot of stress. And actually, the only drug approved for ALS that has any impact at all on in slowing disease progression is a drug called riluzole, which is, and what it does is it enhances removal of glutamate from the synapse. So it turns out that one important function of astrocytes is to remove glutamate, because that's very important, because when a synapse is activated, glutamate's released from the presynaptic terminal, and then it activates the receptors postsynaptically. It's important to remove that glutamate from a functional standpoint so that then the system is ready, gets reset, so it can then receive another stimulus. So, I mean, the scientific rationale for this and using these cells that'll turn into astrocytes, I think, is very sound. Actually, Mahendra, I've written 2 books since I retired. One on the fasting, intermittent fasting, but the second one,
which I have a copy
really?
Of, by the way. So.
Okay. The second one, though, is the most exciting one, no pun intended. And it's at the publisher now. It's called Sculptor and Destroyer, Tales of Glutamate, the brain's most important neurotransmitter. And so
I should look at that. I hadn't realized that you'd written that book, but I will look it up.
No, it's not out yet. It's at the, yeah, I'll send you a copy one.
So I have to tell you about intermittent fasting, though. My wife tried it and it worked quite well for her. And
oh, good.
It's now become a model system that people are trying to do to use in culture to find assessment. So they feed intermittently just to see whether they can challenge the stress response. So that becomes really interesting that you can stress the cells by intermittent fasting.
Yeah.
Changing the glucose levels in the...
Yeah.
So they can look at the pathways activated. So it's been a nice...
Well, maybe combining stem cell therapy with intermittent fasting. Now, there's another, so at this point, we've just been talking about as therapy, transplanting in some sort of restricted cell, whether it's a mesenchymal stem cell for some damage, whatever, to a knee or tendon or so on, or a glial restricted precursor to replace, or not, sorry, our neuron restricted precursor. But there's another approach that is potentially very interesting, and that's so it turns out in the human brain, there are as many astrocytes in the brain as there are neurons, about 100 billion, okay? And so every neuron is kind of associated with one or more astrocytes. And so many people have thought about this. I actually thought about this and had a graduate student work on this for a while with no positive results. But the idea was to take astrocytes and convert them directly into neurons, in which case you don't have to go back through all this things you have to do with iPSC. But moreover, since investigators are identifying certain genes that if you turn them on, maybe even just 3 or 4 genes, you can cause some cell to become a pluripotent cell, or maybe to even become a neuron from an astrocyte. So the idea is that you would use, and typically the way this would be done in vivo. So the idea is in vivo, you directly, in say a patient with Alzheimer's who's lost some neurons in the hippocampus, you directly convert astrocytes, which are still alive in the hippocampus, to neurons, and then maybe, so this is called differentiation. You want to talk about that and what's going on with that? We had 0 success in converting an astrocyte in culture directly into a neuron.
So it's so this has been actually was a very exciting idea, and it was very logical if you think about it, because this had been known even before Shinya Yamanaka showed that you could make pluripotent stem cells from it by showing that a single transcription factor or a master regulator could change the fate of cells. And this was done early on when they took smooth muscle cells and made them into skeletal muscle with MyoD as a single transcription factor, and that was enough to be able to do it. And so then people got very excited when they looked at the iPSC landscape to ask, what do these genes do? And it turns out that KLF and MYC seem to be they seem to be generic in that they help get the cells kick-started, and KLF helps sort of get all the epigenetic profiles modified enough so that you're open. And so it became a very simple idea that we should be able to trans-differentiate into anything as long as we have the right, we use MYC and KLF perhaps, and along with that use some kind of master transcription factor like MyoD for skeletal muscle, etc. And this has turned out to be successful in several limited areas. So turning hepatocytes into pancreatic islet cells has worked quite well. Changing retinal pigment epithelium to retinal progenitors in the eye has worked quite well. People have been trying to do cardiac cells from the cardiac mesenchyme, which is the mesenchymal stem cell, and that has worked well. So here are the 2 problems that have still not allowed us to use this as a therapy. So the logic is very clear. What you pointed out is very clear, that the cells are already in a local place. We should be able to do this in a reasonable amount. And that is that our efficiency of that trans-differentiation process is not good enough. So we don't get really huge success. And the cells which are this intermediate don't just die. They go and do strange things. So you have this problem that you've got this intermediate. If you don't have good efficiency, what are we doing? Maybe we'll cause some damage there as well to be able to do it. And the second thing is the results have been somewhat variable for people, right? For anything which you want to start doing in the clinic, you want it to be consistent, right? So to do that. Nevertheless, I don't want to sound like this is, there are now companies which have said that they have gotten consistency enough that they are going to move forward with clinical trials on trans-differentiation. In the CNS, it is to take the astrocytes and make them dopaminergic neurons right in the part of the brain where you are missing dopaminergic neurons as an alternative way to get to therapy that they're doing to transplant. And in the eye, turning retinal pigment epithelium into retinal progenitor cells so that the rods and cones can be treated has become an important goal for clinical trials. The unfortunate part of this is that when companies do it, they don't publish like academic scientists like you or me would have done. And so we don't know enough about all of this in the literature as we would have liked to know. But it's a very exciting idea. And I often, when I give talks, present it as this would make all of these manufacturing problems and this cost thing that I talk about obsolete, right? I mean, it would not be a problem anymore because we don't need to do it.
And the approach technically is that there's a virus called AAV, adeno-associated virus, and it's really a good tool for expressing genes of interest or manipulating genes. I'm working with a former postdoc on an article now on using gene editing for Huntington's disease.
Yes.
But anyway, this AAV, it's a very safe viral vector, and it's been used in clinical trials for certain disorders. And there are different forms, if you will, of this virus that seem to kind of target certain cell types better. So, in theory, you could have AAV with whatever genes convert astrocytes to neurons, and then you just inject the virus into the brain, and then everything else goes from there. But yeah,
So I can tell you a little bit about this, Mark, because this is something we've been quite interested in. And if you remember, the NIH had a big program with a company called the Horizon, which has been a leader in a lot of this AV work. So the one big problem occurred, which was solved recently, is the substrains of virus which affect different cell types. And for the CNS, we got misled because what worked in mouse was not what worked in human. And it turned out was a different strain. But now that's been worked out as well. But for this trans differentiation process in particular, there's a problem with AAV. It activates the innate immune response, which inhibits this trans differentiation process. So we have this little catch-22 in the trans differentiation piece. But you're right about all the other stuff that you talked about, in the sense that AV8 and AV9, these new seroisoforms, seem to be very good. And AV8 in particular seems to be specific for astrocytes. So we thought that that would be the good way to go for transdifferentiation to occur. But because of this innate immunity response, is that transdifferentiation process has not been as effective as we hoped with AAV. But there's another new class of virus that people have been using called Norovirus. And Norovirus seems to be equally safe and be a better delivery agent as well. So you may want to look that up in being able to do it. And then there's an RNA-based virus called Sendai virus that seems to be also quite useful in being able to do that. And that's already being used in clinical therapy for delivery. And as an RNA virus, you have less worry about integration issues.
Yeah, Sendai virus, Sendai, Japan.
Yes.
Discovered. Yeah. So norovirus is, what are the, you'll hear in the news certain, cruise ship passengers having norovirus or something. I mean, this is a virus that can cause disease, at least it's usually not fatal, but some sort of diseases, right?
Yeah, so it's a little bit like adeno, right? So adeno is relatively harmless, but it can cause a small amount of sort of respiratory type disease, right? So norovirus is a little bit like that, and most people develop an immune response and they're fine with it, right? And the way we make all of these viruses, when we use them for packaging material or delivery, we take out the key genes which make them unsafe and use them as an attenuated version for being able to do these sorts of therapies. So
yeah,
it's like we use HIV now, right, as a packaging thing to be able to use it. It can cause disease, but once we know enough.
Yeah, our rabies virus.
Yeah.
There was an investigator in my laboratory at NIH, Henriette von Prague, from Prague, and she was using rabies virus in some of her studies. It's really interesting. She was looking at stem cells in the hippocampus. These cells in all of our brains are there, and they're capable of forming new neurons, at least one particular type of neuron. And she was using the rabies is
transsynaptic spread.
Yeah, it goes transsynaptic, so you can inject it and it'll go across synapse to the next neuron. So she could figure out when these stem cells give rise to new neurons, what connections do those newly generated neurons establish? And yeah, so there's a lot of value in these different type of viruses and their different properties for basic research and potentially, in innocuous relatively innocuous viruses for the clinic.
Mark, you're right about that. They're also using the same viruses to help do gene editing in vivo, right? So
Yeah.
you can imagine you can do all the correction just like we can do transdifferentiation. You can do the gene editing in vivo and not have to worry about culturing the cells or manufacturing them and getting them all transplanted.
Yeah.
Solve the problem right at the site.
Yeah.
Amazing.
Yeah.
So,
Yeah. Okay. I've been, so I have to ask you, there's like a drawing behind you of a person's face.
yeah,
Who is that?
that's myself actually. So if you look, if it's a cartoon image of myself, so it turns
Oh,
out,
it is.
yeah, it's glasses.
Uh, okay.
Right. So
Yeah.
it's I use it as a part of the logo sometimes when we're doing Zoom meetings because you lose audio. You just want to use a screen when you're talking to be able to do that. And
Ah,
it was something that was done by a Japanese company. I was on the board of the company, and they apparently do this on cards and calling cards to be able to use that. So I just thought this is an easy way to be able to make sure that people know who they're talking to if they're.
yeah. Um, to finish up, why don't you talk about what you think are the most exciting areas going on now in stem cell research in general, and particularly for uh, the nervous system? I mean, I'm asking you like a very broad question, but can you pick out a few things you're most interested in now?
So, to me, the single biggest thing was that there were these two major problems with being able to use cells for therapy, right? It didn't matter whether it was CNS or otherwise. One was solving the immune system issue, right? Or making it so that it was cheaper so that you could use the same person's cells, right? To do all of this. So that to me was a really huge issue. And for the regulators, the big issue was how do we make sure that we don't have contaminating pluripotent cells that are there? And luckily for us, scientists have just come up with really nice breakthroughs on being able to do both of these things. So there was an idea, suicide genes were already in place, but the idea that you can keep them and they don't get lost and the body doesn't silence them, we needed to solve that problem. And Dr. Andras Nash had this idea that he could use something he calls fail-safe, which actually is what we license as a company at Pencella that can be done. So but there are other methods as well, which means that you can kill dividing cells and expect to be able to do it both in vitro and in vivo so that you won't have this issue of getting a tumor late in life or if you're transplanting large number of cells.
Huh.
And the other was this breakthrough initially started by Universal Cell, which said that, look, we can just use these modern gene editing techniques to knock out HLA class 1 and class 2 and do certain further modifications so that you could make universal cells for therapy. And that has really become very possible now. And people have already shown that this can be done, and they've transplanted into humans, showing that if you did that, that you could tolerate this allo transplant, right? So that's a huge breakthrough. And I just predict that if this really holds up and we don't have any unexpected problems with any new technology, that this will just change therapy, And so I think of those as two really important things. And both of them were possible because of this modern gene editing, right? Whether it's CRISPR or whether it's the next generation of prime editing and base editing and, Cas-Clovis and dualase and all of these new enzymes that people have discovered, it's just really changed the efficiency at a scale that now we can do gene editing in vivo. So I really think that this combination has just changed everything.
Yeah. And there's one thing we missed, and I've seen you talk about this in one of your lectures. I think I went on YouTube actually and looked at a couple of your lectures, at one back in India. And so we've been talking about taking certain restricted stem cells and transplanting them or causing transdifferentiation. Now, this doesn't apply to the brain, but it's also possible to make an entire organ in the laboratory, a heart, maybe liver. You mentioned pancreatic beta cells, maybe pancreas. I don't know. Can you talk about approaches to do that? Because there's been some success and certainly advances in this You
So
know, so the idea would be just transplant a whole organ if it's an organ that is easily transplantable.
no, so there's been a lot of progress in that, especially because of the idea of automated tissue printing and 3D printing. So because we can make what we call bioinks, so things which will carry living cells, we can now really print out organs and we can load them with cells. And there was another breakthrough which was called decellularization, recellularization. So where we could take a preformed organ, even from an animal, and we could kill off all the cells and then repopulate it with human cells. And that has allowed us to make nice 3D structures and organs, which we can then repopulate with cells to be able to do this. And now there are companies which are doing this, or there are universities which do this on an individual basis. A good example of that is Wake Forest, which has been working a lot on doing it for the bladder, the urethra, the uterus, etc. And they've shown that this synthetic organ now can function as a full replacement to be able to do that. There's a company called Humacyte, which is making blood vessels so that you can use it as shunts or valves that you can use to be able to do it. And there's a company in Sweden, which is doing this as 3D printing of bone, which they can then reload with cells to be able to do that. And in the CNS, we've now can make what are called mini brains. So, and really, that's just, I think it's just hype. But the bottom line is you can make cells in the right ratio as would be present in the brain. So that the complex of neuron networks, and there's astrocytes, and there's oligodendrocytes, and there's myelination. And as you know, the brain, from all that you've done, you can rewire networks. So by adding a piece of brain, you can allow the brain to then reform it into the appropriate connections or be able to use the connections that are there in a novel way. That's how we repair ourselves after stroke, right? It's by reorganization rather than repair. So you can imagine that in spinal cord injury, you can put this complex and get much better regrowth and regeneration than you could get otherwise. And there are companies now starting to do that. And so I think it's a really important highlight. I mean, it's these sorts of things, as well as the whole large-scale data analysis, which has allowed us to move things forward at a speed that I just not anticipated. So it's very exciting.
Yeah, it's very exciting. When you and I first started, it was,
Yes, it's
you know, if you think about it, it's been very rapid progress in the last 30 years, and it's accelerated and very recently. And a lot of these new technical hurdles that have been crossed that are advancing it. So yeah, very exciting area. It's good to see you. I haven't seen you in years, actually.
It's really nice to see you too, and I look forward to reading your book. And now you should write another book on all these new advances after glutamate, right? So.
We'll see. Right now, I'm doing these podcasts, which they're really intended for say undergraduates interested in neuroscience research, but above, you know. So I'm talking with experts, people that like the forefront of their field. And I think this would be valuable, particularly for students. They can see in one place, it's going to be a lot of very prominent people in neuroscience talking about their research. And it's fun for me because it helps me keep up on what's going on since I retired.
It's very nice. Really good to see you, Mark, and we should meet sometime.
Yeah, we should. Yeah, let me know if you're ever coming to the Baltimore area.
Will do.
Okay, man.
Bye.
Bye.